So hello, this is Oliver Gao. I'm the director of Cornell System Engineering Program, and this is our conversation on systems. Today our guest is Professor Jeffrey Varner, who is a professor in the Robert Frederick Smith School of Chemical and Biomolecular Engineering at Cornell University. So welcome, Jeff.
Thank you, Oliver.
Jeff, it's really great to have you to be with us. So would you like to start by kind of telling us more about yourself?
Okay, so, I started out at Purdue University, and I have an undergraduate in chemistry, actually, and then a master's and a Ph.D. in chemical engineering. My advisor is a applied mathematician within chemical engineering named Doraiswamy Ramkrishna, so we did a lot of modeling and systems work, mostly systems identification, some control work. So my co-advisor is Frank Doyle, who's now is the dean at Harvard, and was at the SEAS, their applied science School of Engineering and Applied Science. So I studied how, and this is work that we've studied biological networks. So all these reaction systems that are occurring inside cells, and how do we understand what they're doing, but how do we exploit them for some technological benefit? So how do I make an antibiotic more of an antibiotic, or how do I produce a protein that we can use as a medicine, or in laundry detergents, so all kinds of different applications. So then I continued with a postdoc at the ETH in Switzerland in the Department of Biology, and studied under another chemical engineer who was in biology there, Jay Bailey. And I then came back to the States, worked at what's now Genencor-DuPont for 5 years, and then I'm at Cornell. So at DuPont, I worked in both bioproducts as well as human health. So we worked in manufacturing proteins for industrial uses, like food uses or detergents primarily, and then worked also on protein engineering and pharmacology for some protein therapeutics that we were developing in the later time in my time at Genencor, or DuPont Genencor.
I see that at Cornell, and your lab is developing physiochemical modeling tools to rationally reprogram human signal transduction and architectures.
Yeah.
Right. So in that sense, and also I noticed that in your talk today, you are going to be talking about cellular, at the cellular level, like the cell interactions. Mm-hmm. So how, starting as a chemical engineer or chemist, how did you move to this cell level research?
Yeah, so this is an interesting, and I'm going to talk a little bit about this or touch a little bit about this. So the origin story of chemical engineers and biology goes way back, all the way back to the First World War, with Weizmann producing butanol and clostridium in the UK to try to produce fuel alcohols because the Germans had blockaded the British Isles. If you fast forward, 50 years, chemical engineers have long been involved in production of biomolecules. So we think about this, or at least traditionally we used to think about this as, well, we're very good at scaling up chemistry. So can we scale up chemistry, but it's occurring inside cells? If you then go a little bit further and think about, what happened when we started to be able to manipulate the genetic material in cells, then chemical engineers came into this field and created a field called metabolic engineering. And Jay Bailey, who was my postdoc advisor, was the creator of this field. And he actually, he's an interesting fellow, because he actually started out as Shell, so in petrochemical, which is a traditional employment area for chemical engineers. But he looked at all the things that they were doing at Shell to control chemistry and said, okay, can I do that but inside of a cell? So his vision, which launched sort of a revolution, was that we should be able to control the chemistry that is occurring inside the cell and rewire all that chemistry to make products of interest. Okay, so now how did I get involved? Well, just like when we are trying to do this in a refinery or another chemical system, we chemical engineers rely heavily on mathematical modeling and quantitation, and so this was a natural extension of our traditional training, but into chemistry that's occurring inside cells. So that's how I got involved in building models of cells. The application areas have just sort of come a lot through some through my time in industry. I was involved in, as I mentioned, at the very end of my time in preclinical pharmacology. So I got very involved in human health and how cells in disease states interact, and why they're in disease states was always a question. So we approached this by trying to understand, well, what's the normal behavior, and then where can this system fail? Because, for example, cancers, I'm painting with the broad brush, but some subtypes of let's say breast cancer, HER2 positive breast cancers, these are is actually a sensor malfunction, because there's a sensor on the outer membrane that's supposed to intercept growth factor signals, and it's locked on without the signal. So we've got a malfunctioning sensor. It's telling the cell, grow, 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 and it shouldn't be grow, 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 grow. So given the complexity of all of the machinery involved, and there's all kinds of feedback, and you have to do modeling to understand how to, if I wanted to steer a cell, if I wanted to control behavior exactly, so I wanted to rationally do it, we need models to tell us how to do it, because it's just too complicated to sit down and figure it out on your own.
I think that's amazing. It's really important, and I totally agree with you that I think it should, because nowadays a lot of people are talking about big data.
Yeah.
And a lot of people are talking about sensor networks, and they feel, wow, we're now entering into this data-enabled, like learning age, such that there are people who are trying to downsize the importance of modeling. However, I do agree with you. It's at the most, it's going to be a hybrid combination of the two approaches together that work better. But it's not really about, okay, I pick up this one and I threw the other way.
Yeah. I've been sort of wrestling with the big data thing, and I'm embracing the big data because it does allow me to build better models. But in the end, so I'm a little bit, when I say models, because I come from a physical background, I'm thinking about, this protein physically binds this one, which does this, which does that. And so I'm really at the very molecular level trying to understand what's going on. The advantage that big data has is, I can look at causal relationships in the data, and I don't necessarily have to know all of the minutiae about how a signal gets from one place in the cell to another. The downside of big data, so that's a real advantage, but the downside is I need it to be actionable. So, when I'm designing a drug, I need to know, well, what protein do I specifically need to modulate, or which state of that protein do I specifically need to modulate? And so I think that's where the models can interface and work well with the data.
Exactly. And also I feel like when you're saying the people, I myself, I like big data, but I sometimes have difficulty so many people are talking about it, what do they really mean? So that's right. For example, even when you talk about, big data, and even when we ask a question, how the big data is created, how the data is collected, and how, for example, do you collect data in an intelligent way? That actually, I think, the help from models.
Yeah, exactly.
Will be critical. And now coming back, I think you are looking at things at the molecular and the cell level. So you're an expert in this area. Can you tell us how cells interact with their surroundings, and how do they monitor and interact among themselves?
Okay. So they interact with their world around them, and they sense the world around them through both chemical means. So there are materials that are secreted by one cell, and so signals sent by one cell through the extracellular matrix that are intercepted, or through the bloodstream or whatever it is, through some medium that are intercepted by another cell by a surface level receptor. So a signal gets sent out, a cell then can sense that chemical signal. So there's a chemical means of signaling, but there's also a mechanical means. So cells can sense their mechanical environment, so how stiff they are, what shape they are, and they have systems to monitor this on inside the cell. So if we go back to, for example, the receptor, if I have receptors that are activated on the surface, then you'll get a whole series of events that will occur that will lead to some outcome. So the classic one in growth factor signaling I'd mentioned was, well, I've got a growth factor, the receptor comes together, it dimerizes, and then there's a phosphorylation cascade. So a classic one's called MAPK, mitogen activated protein kinase cascade, that senses growth factors. And there can be both transient signals, so I can transiently sense the growth factor, or I can also lock a system on. In a similar way, there are mechanical transducers on the membrane that turn a mechanical signal into a chemical one, because the cell inside operates on a chemical basis. So you've got to convert that mechanical into a chemical one. There's also, for example, electrical cells can sense a potential field that they're sitting in, for example. And this is done by converting this electrical signal, for example, current flow or ion flow, into a chemical one. So calcium gradients or sodium gradients. So in the end of the day, what we try to do is understand the chemical and then build the different ways of transducing the signals from whatever type they are into the chemical. Yeah, I would say, though, we're just now emerging into trying to understand what's going on outside of the cell, because there's so much fascinating stuff going on just inside the cell itself that this new area of mechanical signal transduction is it's a really important one now, and this is an area that we're getting involved in.
So now I would like for us to jump out the box a little bit. Okay. So you can see that you're talking about the cells and how they interact. When you talk about how cells behave and how they interact, I couldn't help thinking of humans, how we interact, how we with ourselves. But anyway, and then why a system approach? Why do you think system approach is so important for you to look into these kind of cellular level interactions and mechanisms?
Well, this is a sort of a paradigm that we have that is not always shared in the biological world. So the classical way that biology was done, let's say cell biology was done, is that a person would study one particular molecule and all the things that it does, and maybe look one step away in the things that it's interacting with, and then over time, after many lifetimes of researchers, we've built up this sort of complex understanding of what's going on, but still there are a lot of holes in the network. The reason we study it in a systems approach is because everything is occurring at the same time. So it's not just one single process that's occurring, but there is one process in thousands of things that are occurring all at the same time, and they all can talk to each other and they can all interact with each other. So if you don't consider the forest and you only focus on the tree, then you cannot get sort of gross system level behavior. So how a cell decides to differentiate, for example, so change into a different cell. You'll capture a part of it, but you won't capture the overall thing. And if you zoom out, even what we're studying is only a small part of an overall system. And what we do a lot of DoD work, and they refer to this as a system of systems. So I've got a cell that's sitting inside of an organ which is plugged into the circulation. So you can sort of grow out like this, but this new area of mechanical transduction, or a lot of the things, for example, that BME are looking at whole tissue behavior. This is sort of the next step in the evolution of us trying to understand what's going on. We have been focused more on just individual cell actors, but you can think about these, much to your human analogy. These are agents in a large system, and they have rules about how they're reasoning, and so we can then start to simulate collective behavior based on understanding the individual elements in a system. So we do a little bit of this, and this is an area that we're moving into primarily in cancer area. as part of our Physical Science Oncology Center here on campus.
When you talk about kind of how cells they respond or the measure through chemical as well as physical kind of processes or mechanisms, and also just now you were talking about this kind of system of systems, so and also agents, right? Now let's jump out of the cell level and let's jump out to societal level. In our societies, we are facing a lot of challenges, and so as a result, people are now more and more using a word what they call social technical systems, kind of where you have the technical component, you have the human component. That as a result, because the technical component it's fixed, it's an object, and you know how it's going to behave. While the human element, the social element, there is a lot of uncertainty. So now, zoom back to the cellular level, cell level. So you said each cell, they are actually all these cells. They are living also in the community. They are communicating. They interact through physical and chemical kind of these procedures. But from your research, did you also observe any kind of social element equivalent among cells? For example, of course, human beings, we have mood. Right, I assume that, of course, at least I know that when we are in a bad mood, probably in our stomach, we will not function well. So assume that the cells probably will be living in a miserable environment. So is there any social element equivalent at the cell level?
So the way that cells sense the population, and I'm going to focus not on human cells, but we'll go to bacterial systems, so a little bit less complicated. But there's population behavior even amongst, let's say, E. coli cells, and this is done through a system called quorum sensing. So cells can sense the number of cells, the size of a population, and there are programs that are kicked on based on the size of the population, and then the state that population is in. The classic one is called sporulation in Bacillus, where Bacillus is a soil bacteria, and it's interesting that it can undergo a process called sporulation where it will, and I'm kind of painting with a broad brush, but it will essentially grow a hard shell around itself and then hunker down, and it can stay in this dormant state for decades, and then one day it'll pop back up. And this sporulation network is driven in part by cell-to-cell communication, because some signal says, okay, it's time to sporulate. We're either in a nutrient-poor environment, and we want the population to survive, so we're going to go hibernate essentially. In the cancer world and in the mammalian cell biology world, there are a lot of situations where collective behavior is influenced by the actions of single cells. The one that's top of mind right now for some of the things that we've been doing is this in tumors, this thing called a Warburg effect. So cells can make ATP by a couple different energy. They can make energy by a couple different ways. One of them is very efficient, requires oxygen, and the other one is less efficient in the sense that I have to trade some of the carbon, so sugar, that I would use to make more cells. I have to trade it to make energy. So it will work, but it's less efficient. So cancer cells, this has been known for quite some time, even though there's oxygen around, they will use this less efficient route to make energy. And the byproduct of that is they will spit lactate out into the media. And so there are a number of hypotheses as to why they do this, but from a sort of social perspective, the hypothesis that Abe Struck and I, so Abe is a faculty member in chemical engineering, are trying to work out is, does suboptimal behavior on the part of an individual lead to better behavior as a system, as a whole? So, if we were to counterintuitively make the cancer cells produce ATP more rapidly, collectively the tumor wouldn't be as healthy. So it would have a benefit. So this is something that we've been exploring by, again, this agent-based procedures, but it's an intriguing possibility. Off the top of my head, I mean, I think it sounds kind of crazy, but we'll see.
It's really the cellular interaction and actually the agent's behavior and the system's interdependency. And I heard you mention several times you are using, one of the key tools you use is mathematical modeling. Why mathematical modeling in dealing with this at this kind of tiny scale?
I've never actually been answered or asked this question. So I think that it's a little bit how I'm wired, because I tend to be very quantitative and less qualitative reasoning. I want to know, well, how much of something and the rates at which things are occurring, and the tool set that I use to reason in that way is mathematical modeling. Mm-hmm. All different kinds of models, not just the traditional sort of differential equation based models, but it's another tool for me to build a mental picture of what's going on. And I do it in terms of sort of qualitative pictures that biologists generate for me. I make them quantitative and see, well, given the measurements that we have, is that qualitative picture consistent with the measurements that we have? And that helps me understand what's going on. I think it's a little bit, I find it to be a handy tool, but I guess the real answer is it's just how I'm wired. It's completely natural for me to think in that way.
I appreciate that there are people like you, thinking about problems in this kind of wired way, because, from qualitative, which is, of course, is very important to give us a big picture. However, big picture could have loopholes. Yeah. And I think the beauty of math, it helps us for our thoughts to be more complete and more rigorous. So, right, because when you try to derive equation, right, if you cannot derive it through, right, it doesn't make sense, right? Yeah. So you have to just, get that through. So speaking of that, and you earlier mentioned this kind of, suboptimal cell level behavior could lead to system level optimal performance. And this is kind of definitely reminds me of, this year's economic Nobel laureate, Nobel economics prize went to behavior economist, mm-hmm. right? The behavior economics, right? It's kind of totally, it's exact kind of on the contrary of the the traditional economics, where we assume that each agent, they're acting rationally, they're trying to maximize their own utility. Mm. So that kind of goes back to the question. So at the cell level, do you think that each cell is acting in a way that they're maximizing their own, for example, not necessarily utility, however, kind of their own performance or benefit? Or do you think that cells are more altruistic than human as a whole?
Well, there's actually been a lot of work done on this. And so I can give a semi-definite answer here. At the level of bacteria, so E. coli, bacillus, yeast, so these simpler cells, yeast is a eukaryote, but let's lump it in with the bacterial group. There's been a lot of work done to show, or to get at, what are the objectives of a cell? What's it trying to do? So it takes up glucose, takes up nutrients, and it's growing and doing its thing, but what is it actually trying to do? And there's been a fair amount of experimental work as well as theoretical work that shows, at least for simple bacteria, so the E. coli or bacillus, for example, that the cells try to maximize their own growth. And they do it at the expense of their neighbor.
Wow.
Meaning they try to consume as much sugar as they possibly can, and they don't care about their neighbor. They could starve their neighbor. They don't care. And there's all kinds of other things in the like secondary metabolites in the in the bacterial world, where we have multiple different species, and there's a lot of chemical warfare that occurs in the bacterial world, where they'll try to poison their neighbors so they can get the lion's share of the nutrients. So in these simpler organisms, there's both experimental as well as theoretical work that has shown that these cells do not act. They act selfishly. In the mammalian world, so in more complex biology, it's a little bit less clear. It's not true because of the sophistication of the cells and they're in different states. It's not true that cells act to always maximize their own growth. So that would be our measurement of utility or benefit. Traditionally, cancer cells have been thought to maximize their own growth, but they could actually grow faster if they didn't spit out lactate. So if they were using this higher higher efficiency way of generating energy, they would have more energy and more carbon, which we would presume would lead to a faster growth rate. But they're more complicated than that because they think bacteria you can think about as sort of survival of the fittest, but in the mammalian world they're not wired that way because there isn't sort of a culture, if you could use that phrase, of being a cell on its own. They're always part of a collective. So I think that in that case they do act more for the societal benefit and less for their individual benefit.
Wow, this is really interesting to know. Like, of course, you're talking about probably in kind of earlier species versus kind of more advanced species. Mm-hmm. There seems to be some evolution or difference.
Yeah. Of
course, that kind of the easiest way I can think of is that the earlier species, like kind of their brain system, their nerve system, is not as advanced as later species have. So what caused that change? Is that due to, for example, this kind of selfish activity pattern? Is there even any variability of this selfish activity between the brain cells and the muscle cells? Because I feel that if the brain cells, they are acting less selfishly, so that they are probably acting as something.
Well, I'm sure there's cell-to-cell variation. Mm-hmm. I mean, the one difference is though that these between bacteria and our types of cells is that our cells will exist in tissues, and so that means by definition they're stuck to a neighbor, or they're lining a blood vessel or something like this. So they are always interacting with another cell of similar or of the same type or similar types or different types. For example, cells lining the blood vessels will be interacting with all the types of cells that are in your blood. Mm-hmm. But in a bacteria, there are examples of collective behavior in bacteria. So some cells will form biofilms, so they'll grow on a surface. But traditionally, that's done under certain nutrient conditions, and so that trips a program where it's more of a survival thing as opposed to being something that they would do on their own. Traditionally, these bacterial cells, they don't interact with one another. They do communicate via quorum sensing, so they can sense how many cells are around, and they can communicate through the nutrient environment, so through the amount of nutrients that they're in their world, but they don't interact in the same way that mammalian systems do. So mammalian systems, for example, have all kinds of different proteins on their surface that lock them together and stick them to things, and this is that's a program that they have, and you can turn that program off. Often in cancer, so cells that line blood vessels, that program will get disrupted and they will then just become detached. But if you were to leave everything alone and everything were fine, they always act in a collective way. So whether a nerve cell and a muscle cell or a cell lining a blood vessel, they're all going to behave differently, but they will all sort of share the same collective behavior in the sense that they have to interact with the cells around them.
Yeah, speaking of this, and then in connection with the mathematical modeling approach that a lot of us have been using in our research. Like when we do mathematical modeling or optimization modeling, we always have objective function. Yeah. And we have constraints, and we try to solve the things. But a very fundamental thing we say that we assume that this agent has an objective function that it is trying to pursue.
Yeah.
However, we look at this kind of cell behavior, and even look at in our society, there are many people, it's not necessary that the cells, they're acting to maximize the objective function. Instead, they're probably just living their lives following certain principles. As long as it's within the principle, they will just live naturally. But after the principle, they'll probably have some adjustment. But do you really believe that? Because you can see when optimization is one powerful tool that a lot of scientists are using. But do you really believe that the behavior of a lot of agents from cell to humans, do you really believe that they are acting in pursuit of a specific objective? Because even for us, from time to time, we don't even know what objective we have in mind. So is that kind of goal-seeking behavior, or is that kind of principle-guided living?
Well, at the cellular level, there's actually been a fair amount of work done here thinking about that cells can operate with respect to multiple objectives. So they may have multiple objectives, but it's always an objective-based goal-seeking behavior, at least in the bacterial world. So I need to differentiate that from mammalian systems. But in the bacterial world, there's been a fair amount of experimental evidence to show, for example, that I can take an E. coli cell and grow it up on sugar 1, and keep it in sugar 1 for a long time. So it becomes accustomed to sugar 1. Then I can take it and I can put it in sugar 2. So there's no more sugar 1. It's got to adapt. And I can predict, just based on the biochemistry, what the maximum growth rate that this cell could have. And if you wait long enough, it will go and it will achieve that growth rate. And so this was a paper by a researcher named Bernard Paulson. It was in Nature, I think it was in 2004, that showed, it was the evidence that one showed that a maximum growth rate was a reasonable objective for these types of cells, and that the cells will adapt their machinery will become directed, self-directed. Now, since that work, more sophisticated cells, so yeast and up, we think that they're not looking just to grow, but they're looking to do many different things at the same time. But they're always trying to do, or at least the way the community describes them, they're always trying to achieve a suite of possible objectives. So this, I don't know what it would mean be principle driven in like a E. coli cell. I don't understand what that would mean.
Yeah.
So, because they're dumb. The only thing they can do is grow, right? And we take advantage of that.
So that's right. Which means that, for example, right? If I'm bite by a bug, and even without any kind of goal-seeking process in my mind, I just So you can say this kind of reaction, it's not goal-seeking. It's just you bite me. It's kind of basic instinct.
Yeah. Mm-hmm.
So of course, I probably kind of underneath basic instinct that might be kind of goal-seeking.
Well, it's a response, right? So you feel the pain, and then
That's right.
So it's a learned response. If I could have put a video camera on you for as long, from day one to now, at some point, perhaps you were bitten by a bug and didn't swat, but you learned because it hurt. So, but that's an interesting way of thinking about it. I don't know what that would mean though in terms of an E. coli cell.
So now I even more kind of out of the box thinking. I'm really amazed because when you are describing the behavior of cells, like if you don't call it a cell, if you just call it an agent, and people, when they listen to what you're describing, they're probably thinking, oh, Jeff is talking about our society.
Yeah. Mm-hmm.
Right? So in that sense, it's really kind of, I think that's actually, one of the key things is really the keyword scale and granularity.
Mm-hmm.
It really depends on what scale and what granularity we're looking at things. So, right, you are doing research at the cell level. You are indulging yourself, looking at finding the beauty and understanding their behavior in your lab, right? And when you get out of your lab, you are living in our society, right? How does your research and understanding of this kind of cell level behavior interaction, how does that change or affect your view about our societal life?
I mean, they're kind of far apart.
That's right. That's why I was saying that in the more out of box thinking.
Yeah. So, well, I mean, it does inform me in a certain, in a number of ways, actually. So in kind of ways that, for example, when I'm at the at Costco or at BJ's or whatever, and I see Tide, the laundry detergent. Well, I know the organism that was used to produce the active agents in that Tide. I know how that enzymes were formulated in that Tide. I know all of the thought that goes into, because it turns out laundry is kind of complicated, but to engineer the proteins to work in that environment. And then if you scale up, in terms of, I understand, probably the one with societal impact that I most feel every day is from the cancer work. So I got involved in cancer because Jay passed away from complications from cancer, and it was the first time that it had impacted me directly, because when he got sick, I had to, even though I was still in the state, I was in the States at that time. I flew back, because this was at ETH in Zurich, flew back and taught sections of the course that had to do with math, because he was not able to do it. And so I understand not only at a molecular level, or at least have some insight at a molecular level what's going on. I wouldn't say understand, but I potentially understand what's going on. But then also from treatments, what are the risk rewards for treatments, and what's cutting edge for treatments, and if I could design a treatment today, what would I want it to do? If I could magically design a treatment, what would it be? So I have a better understanding, not to the degree that a clinician has, for example, because I'm not interacting with patients every day. When I was during my time in industry, I did interact. We had a couple different clinical trials, so I did interact with patients there, so I can see, the real consequences of what's happening at a molecular level, but scaled up in a person, and not only the person, but then the people that person interacts with, and the toll that it takes on a family, and so forth. Those all come from these simple molecular malfunctions at the cell level. So it does. I do appreciate a little bit more sort of the network effect for these things.
Yeah, I think with the mathematical modeling tools you have developed, and I think you have tried to apply these, for example, in the context of biotechnology and human health application. And I believe that's actually a very important value creation process for us researchers, so that our research just doesn't end up with a top journal paper. Instead, that could go a longer way to impact the real world. So in that sense, kind of what and how have you been doing in terms of translating your research to the real world application?
Well, this is something I want to do more of. I want to have bigger impact. I mean, we've done a few things. So we do a lot of work on trauma and blood coagulation. So how your body responds in acute, after injury in the first couple hours. And some of our models have been licensed from industry. So the one licensed, probably the for folks that work in the area, is a company called Farsight, which does a lot of PK and PD modeling. So it's one of the FDA-approved software suites to do pharmacokinetic modeling.
Congratulations.
So we've also done we've done a lot of work trying to figure out targets. So addressing, well, if I could hit something inside the cell, what would be the best possible thing to hit? So this, because if I'm going to design something, I want it to have the maximal impact. So we've worked at developing some technologies, that are patented or on their way to being patented to actually experimentally modulate some of these targets that we're going after. And so we're just on the cusp of really being able to show translational benefit there. I think the last thing that we've done is we've had a fair amount of impact trying to understand the data that people are generating clinically, and then marrying that with what's in the literature, both with primarily in the trauma area, but now also more in the cancer area, where we're looking. So, for example, we work with a project in the vet school, and I'm going to talk about this one today, where we are trying to understand at a molecular level how a resistance cells that become resistant to treatment, how does this work? And because if we can figure out how these things work, then and how we can block the resistance, and this could lead to nutritional treatments for cancer, so certain leukemias. So this is where we are having on the potential cusp of having impact. But I'd say primarily right now it's been, we release our models, open source all of our models, anybody can use them, and so we have a lot of people that use it to try to understand what's happening at a molecular level. I mean, the grand vision for us would be to build a mega model of different tissue cell types. So breast cell, so for breast cancer or prostate or leukemia, and then release these to the world so that we could simulate what were different interventions. We're infinity away from that, but we are working on bits and chunks of all the different pathways that are involved. It turns out to be a difficult problem. The scale of the problem is very large, and so, but we're on our way. We're on our way.
Wonderful. What an amazing career you have had. And if we kind of look back, right, you studied as a chemical engineer, right? You studied chemical engineering, and then you of course you did a Ph.D. and you did postdoc, and you work in the real world industry. And then now look at what you're talking about. You're talking about cells. You're talking about cancer. And you're talking about mathematical modeling, computational modeling. I can see that with the domain, like when first you have a very deep domain knowledge in chemical engineering, but now you can see you have a very wide knowledge structure that enable you to be able to do all the things. Yeah. You have, I think that's really a very good reflection of what we call like a T-shaped knowledge structure, right? You have the roots in the domain knowledge, in the domain area that you were educated, trained, but now like a lot of things you're using is really as this horizontal bar where you have this knowledge structure. So in this sense, what would you say to about education of our future, not only engineers but also scientists? Because you can see that apparently the challenge we're facing today is becoming more and more complex systems in nature. So what would you say to like future generations of engineers in terms seeking their education and also their career development?
Yeah, so I would say that one of the things that has made me successful is a deep appreciation for fundamentals. And so not so much focused on applications per se, but more trying to understand fundamentals of processes, fundamentals of physics and thermodynamics and kinetics, and these are important things in my world. I think all of these different problem areas that we work on, in my mind's eye, they're exactly the same problem, which is I've got a reaction system. I've got a bunch of proteins or a bunch of small molecules that are being chemically converted to something else. That I've got inputs and I've got outputs, and things change in time, and there's control that over top of it. But that's exactly the same problem just repeated over and over and over again. And so I've learned tools and techniques to kind of take these fundamentals and apply them. The other area the other thing I'd suggest is, especially for modeling people, if you want to work in the computational area, do some experiments a few times in your life, because in experimental data, the devil's in the details, and there's all kinds of little intricacies in the data. And I went through a period where I, in fact, going into my Ph.D., I was all experimental. So I didn't do a lot of modeling work until I discovered one day, I'm like, man, there must be a better way of thinking about this problem. And that's when a light bulb in my head, well, why can't I simulate this? And then instead of me having the experiment, so it's sort of the tail wagging the dog. So instead of me doing experiments to build a model, let me build a model and then figure out what experiments I need to run. And then that insight, because then I said, well, I don't need to run 20 experiments. I just need to run like 2 best ones. And so that completely changed the way my sort of worldview. But I still do appreciate, because I every day work with experimental folks. That's sort of the thing that we, to address, the world becoming a more interdisciplinary place. So my group over time, we work with a lot of different folks at Cornell, and then throughout the country, clinicians, other modeling types, basic scientists, and we have almost become like a service center. We have a very particular skill, and we have built, because we understand how the experiments are run, at least in some cases, a very deep level, but in other cases, a little bit perhaps more surface level, but we can communicate. We've built interfaces with these experimental types. We can bring value to a number of different teams because we understand their language and we kind of understand what they're doing.
I think it's a very great, kind of recommendation, advice to the student that kind of having a very deep appreciation of the fundamentals. I think that's definitely very important. And I think, unfortunately, we're running out of time. I think I need to move you to another room for your great presentation. But, Jeff, it was great having you.
Thank you very much.
Thank you. I think we're all set. We are moving to another room.